Hello, everyone. My name is James Green, and today I'm going to be presenting my project, which is gene annotation of the MELT gene in Drosophila species. A little bit of background about me. Prior to the pandemic, I was working with CRISPR and E. coli, doing some mutagenesis and genetic recombination research. And it was really rewarding. It was fun to be in lab, to come in at, every day and do some research, check plates, inoculate cultures. It was a lot of fun. I learned a lot. But when the pandemic hit, I could no longer come to campus, and that was unfortunate. But I still needed to, do research. I still needed to produce and contribute to the community of science. And so when we switched my project into gene annotation, I was introduced to it by my late mentor, Dr. Jim Youngblum, and he showed me this project from the Washington University of St. Louis called the Pathways Project, which is run by Dr. Laura Reed from the university. And one of the things they do is they annotate genes, and what that is they look at the unique characteristics of genes within different species. So Drosophila melanogaster is the most studied species of fruit fly, and we've sequenced its entire genome, so we know just about everything there is about how its DNA works and how its genes works. But we don't know as much about its related species, the other species in the genus. So what annotation does is it looks at specific features like the reading frames, characteristics of introns and exons, introns being the regions of the genes that are spliced out, and exons being the maintained and translated and transcribed portions of those genes that result in the traits we see when we look at ourselves or we look at the fruit flies. And I focused on annotating the MELT gene of 7 different Drosophila species. And my goal, a little background about the MELT gene before we go to my goals of this project. The MELT gene is involved in the insulin creating pathway. The way it's involved is that it modulates a couple transcription factors that increase or decrease the production of insulin. Primarily, its role is in insulin production. So whenever it's expressed, it causes those other promoters to be expressed and recruits receptors to the cell membrane, allowing insulin to be created. And so what we see as a result of that in adult fruit flies with mutations in this gene is that they end up having less body fat. Than normal, than average species. And also, during development, it also plays a role in the rhodopsin expression or production, sorry. And what rhodopsin is the actual structures, the pigments within the eyes of the fruit fly that allow them to see different, wavelengths of color. We see visible light. Flies mostly see visible light as well, but some other frequencies. And those frequencies can be changed based on the production of rhodopsin and specific rhodopsin pigments. And so my hypothesis in this experiment was looking at the relatedness of the MELT gene to the parent species of Drosophila melanogaster. And so here I have a few images that are showing the relatedness of different species. Drosophila jacoba right here is going to be the closest, in theory, the closest related species to Drosophila melanogaster because it is a very close relative. And then we have Drosophila persimilis, which is intermediate. And then we have Drosophila pseudoobscura on that side, and it's more distantly related. So the trend should be from higher to lower similarity within the MELT gene as compared to Drosophila melanogaster. And so how I went about doing that is first I had to make the order. I didn't make the order, excuse me. First I had to find the order of similarity between these species that I looked at and melanogaster. So at the top here, this tree, this phylogenetic tree, we can see Drosophila melanogaster, which is what we're comparing everything to. And then below that, you can see the different species, and the lines that are connecting them are showing relative similarity, right? So anything closer to melanogaster is going to be very similar. As you can see, Yacuba and melanogaster have a pretty recent common ancestor as compared to the other ones on here. This is not all the ones I study. I will have a phylogenetic tree in the following slide. Actually, let's go to that now. But as you can see, as we go down the tree, as you can see on that side, we'll go from more similar, which is closer to melanogaster at the top, to less similar as we move down the tree. And on this page, we have the tree that I actually based my order of similarity on, because this is the most consistent data. I found some conflicting trees that showed on the side over here to this way. You have Virulus and Grimshawi, which I sampled, and what we can see is that sometimes the order of those is switched, which would basically change their relation to melanogaster. But this is how I based it, and above me on this side is how it is in the genome browser, which I'll go over in a moment. And so my methods for this experiment were to, this is the basic workflow of the experiment. And so starting at the top left, we locate the MELT gene. That can be done by a simple search in the genome browser for the MELT gene, or more accurately, it can be determined by taking the BLAST sequence of the Drosophila melanogaster MELT gene, putting it into the genome browser, and basically the browser will tell us, hey, right in this area, that's where we got the closest alignment. Then after we have the general area, we got to note the genes that are surrounding the gene in question. So this, the MELT gene, I have to find what's upstream, what's downstream, and take note of that. Then I got to find where the start codon is, basically where transcription of the gene begins. And then right here, following the arrows, I have to note the coordinates for the start and end of each exon. Like I said earlier, exons are what actually code for the protein products that we see in the organisms that we study. Introns, which are in between these exons, get spliced out during splicing, which is a normal part of transcription and then translation. Then I use a program that the Genomics Education Partnership maintains. It's called the Gene Model Checker. Basically, I put the coordinates in that. It tells me if I did it in the correct format. And then from that, I can obtain a couple graphs, or sorry, a couple data visuals and additional data that I can use for my final analysis. And so the genome browser, the Genomics Education Partnership, that's what GEP stands for. Genome browser is based on the sequence data from UC Santa Cruz in collaboration with the Washington University of St. Louis research as well. So basically both their bioinformatics groups collaborated and created this genome browser, and it's maintained by WashU St. Louis. And here's what a screenshot of the genome browser looks like when you first search or use the BLAST data to find the MELT gene. And what we see right here is that the MELT gene is right there in the center. You can see it in purple, or you can see it in red at the top. The red at the top is a BLAST analysis, like I mentioned before. The purple is the SPAL-N alignment, which is a program that has used, bioinformatics, right, to approximate where it thinks the MELT gene lies on Drosophila yacuba in this case. And we can't really trust its alignment. It's great for finding the general area, but we cannot trust it. What we can trust, as we see here, right here, is the adult females and adult males tracks, which are these blue and red bar graph looking data. That data we can trust because that's RNA sequence data. What that means is it's actual gene products that we can measure and quantify. And so when we see nice peaks there, that means that genes are being transcribed and then translated. And so I have to zoom out from that view using the functions at the top there. They have zoom out and zoom in tools, which is great. I zoom out and you can see the MELT gene now is much smaller, right there in the middle. And then to the right of that is the corn gene, and to the right of that again is CG8620. Those genes would be considered downstream because transcription is happening from left to right in this case. And then on the left, we have the ACBP2 and ACBP3 genes. Those would be considered upstream because they happen prior. This is important because genes that are often involved in the same pathway are often near each other on the chromosome. So it's important to take note of these characteristics, although they're not entirely necessary to get the similarities to the parent sequence of melanogaster, but they are important in terms of qualitative analysis. And so to get the coordinates that I talked about before, we actually have to zoom in further. So this is like the view prior. In this view, we actually select the very first exon, which is that thick bar. You know, if you're looking at it's easier to look at the purple sequence. If you're looking at the purple bars right here, the thick bars are exons. The thin little lines between them are introns. Again, introns get taken out. Exons are what we care about. And so this is a zoomed in picture of that first black, thick purple bar. And this is the first exon. So what we need to take note of that, the first exon needs to have the start codon, which starts transcription of the entire gene. And we can see on the left here in this purple circle, and the little zoomed in feature on the left, that's the start codon, methionine. So that's ATG specifically in sequence. So I would basically mark that as my start coordinate. And then I would go to the right here and look for where the RNA sequence data, which is this data right here, these two lines, the red and the blue here, where that data ends, I know that transcription has ended there. So I go up and I look at the coordinate number and I note that, but I first need to check to make sure it has a GT directly following that number, because that GT is called the splice donor, and the splice donor is the beginning of where the splice enzymes would bind and then cut the intron out. Again, thin line are the introns, thicker lines are the exons, and exons are what code for products, introns are removed. And so at the beginning of each intron, there's a GT. At the end of each intron, there's an AG. Those two signify the beginning and end of an intron, and those help the splice enzymes. They're essential for the splice enzymes to remove that sequence. And so once I have all the coordinates, I take them and I list them in a very specific format with the start coordinate, hyphen, end coordinate, and then a comma. And I do that for each exon. In this case, Yakuba has 9 exons, and I have to put those numbers in here. And then when I hit verify right down here, that this result will pop up, not necessarily the first time. This is after a few tries, because whenever you get something a little bit wrong in the format or you don't have the right coordinate, all those like passes or a lot of them will turn red and then tell you kind of what's wrong. But then you have to go back to the genome browser, look at the genome again, and try it, give it one more go until you get all green like this. And at the top here, we see that there's different tabs. There's a dot plot tab and a transcript sequence tab. Those become important to our next analysis. And so within the dot plot tab, you have a dot plot. What the dot plot is it's showing the relatedness to melanogaster on the x-axis to your studied specimen on the y-axis. Basically, wherever they're the same, it's going to be a one-to-one correlation. So you have a nice linear graph going from corner to corner is great. You don't really want gaps, because gaps indicate that they did not match up. And then if it's off and it's not linear and you have an inconsistent chart, basically means that the sequences were different, or they didn't start at the right spot. So we can look at a couple of examples later to show you what a bad one looks like and what a good one looks like. But I may not have time for that in this presentation. And then these little dots that are extra in the middle of that dot plot, they're fine as long as they're symmetrical, because that indicates a correlation between them, but they're not ideal, but they're not detrimental to the data. And so what I'm going to go back to here is another window within that dot plot tab. This is the protein alignment. So this isn't actually showing DNA sequence on that other side. It's showing codon sequences. What those are is the actual protein products that are produced by the DNA sequence. And I made this little graphic up right above me here, showing how to analyze this. Basically, the red and the orange is the Drosophila melanogaster species, and wherever they alternate, that indicates a new exon, right? And then the blue and light blue is the species that you're studying. So in this case, it's Yakuba, which is designated by the DY, and they are compared 1-to-1. Each thing is compared. Each codon is compared directly from the sample to the parent, which is melanogaster. And whenever they're the same, it's indicated by an asterisk, as I've shown there. And if you look really closely in the protein alignment, you see some that don't have an asterisk. That means they're dissimilar and not the same. So this one actually looks really good because Yakuba is, again, very closely related. And so that other tab I mentioned with the transcript sequence here, that has the sequence of the protein alignment, but in a format that I can use in BLAST software. So I would basically copy that and then go to the BLAST analysis, right? I would put that sequence into my query box, and then I would put melanogaster sequence in the subject box and then compare them and then get my results down below me. Now, what I'm looking for is the percent identity, and then I'm looking for a low e-value to show a good sequence run. In this case, it's 98.38% because it's very closely related to Drosophila melanogaster. And now to show all my results, here is what I was not hoping for in my hypothesis, but that's how science goes. We got to correct ourselves and manage that. But here shows all my data from the left to the right, shows the different species and their relative relatedness. So Yakuba is all the way on the left, meaning it's most similar to melanogaster, and all the way on the right is Grimshawi, and that is the least similar, the most distantly related to melanogaster, and they follow in that order of relatedness. On the y-axis, we have the similarity to Drosophila melanogaster, 100% being exactly the same, and then going down in relatedness. And on the x-axis is all my samples. So as we can see here, I was hoping for a nice line trending downward from the left to the right. But unfortunately, according to the data, that is not correct. So this supports the null hypothesis, which is that the gene is, the distantly related species do not experience a negative correlation to relatedness. And so what this shows is that the MELT gene is actually very well conserved in the Drosophila genus, which is understandable and implies further supports that it's essential during development and as well as health, because if the mutations were too great within the MELT gene, then it may result in the fruit fly not surviving to adulthood and therefore reproducing. So the MELT gene is critical, and we can use it as an analog to study insulin production. So I'd like to thank everybody for listening, and I'd like to thank the McNair program very deeply for this honor to be presenting. It's been a great experience doing research and attending conferences when we could, and also having the resources and support is truly amazing. So thank you for listening, and have a good one.